Okay, so welcome all of you, and thank you for joining our office hours, where I will introduce how to explore computed structure models at rcsb.org. So let's start by the beginning. So what is a computed structure model? So don't take this as a formal definition, but I think it's going to be useful for this presentation, and it describes the situation in the context of rcsb.org. So we can define a computer structure model as a protein structure that has been obtained using computational methods, and there is no validation or support from experimental techniques. So currently, the methods that the most accurate methods are based on machine learning, in particular neural networks, and they only need the protein sequence for predicting the structure. This is not completely true. From the protein sequences, they collect multiple sequence alignment, and also they use 3D structure templates. I'm not going to go into details of how these algorithms work, because this would require a complete session for that. But examples of these methods, I am completely sure that all of you have heard about AlphaFold. So the version 2, together with RosettaFold, was the breakthrough in the field of computational protein folding. There are many other methods that they work really well. For those of you that are not familiar what CASP is the critical assessment of protein structure prediction. So these are a series of experiments that happens every 2 years to evaluate protein folding methods. So what happens when a structure that is considered useful for the evaluation of protein methods, so what they do is, instead of releasing the structure, only the sequence is released. And the different participants, what they have to do is to propose potential models for these proteins, and these models are evaluated, and the different methods are scored. So from the point of view of CASP, the problem of predicting single protein chain is considered solved. Of course, that we have to be quite careful here, because this doesn't mean that this method replaces experimental technique, because CASP has a really specific way of how protein structures are evaluated. So here it's considered solved from the CASP point of view. But it's true that these methods work that good that in the recent year, different databases have appeared compiling a structure using this computational model. So for example, AlphaFold DB is a database that what they do is they provide the model, a structure model for each protein in UniProt. So for example, ESM metagenomic atlas, more than an active resort, it was more an experiment using ESM-Fold. They predicted the structure, I think it was from 600 million of metagenomic sequences, showing the scalability of, in this case, ESM-Fold. Sorry about that. So why CSMs are important? Well, if we think about the amount of data that is collected from protein sequences, so there is no way that experimental determination methods can cover all the proteome. So what happens is that many organisms are underrepresented in rcsb.org. So we don't have information from experimental methods for many proteins. Also, if we go one step forward, and instead of thinking only in protein single chain structures, we consider structures for protein assemblies or complexes, structures that are from mutating residues, or that include post-translational modification or protein ligand interaction. These data, the structural information, grows exponentially. Again, we have to be careful, because these methods work really good in single protein structure determination. They don't work as good on this situation. They start to work pretty good with predicting of macromolecular assemblies, protein complexes. But I am completely sure, I'm not sure it's going to happen in 5 years, 10 years, or 100 years, but this method will quite improve in also all these possible situations. And what is going to happen is the amount of information that we'll be able to collect from these methods is going to be massive. There is no way that we can obtain information for all the possible complexes or consider any mutation of translational modification that could happen in any protein using experimental technique. So why they are important in the context of rcsb.org? As I said, the issue is the proteome coverage. If we analyze what is the content of rcsb.org from experimental structure, we can see that they only represent around 70K of UniProt entries, while the UniProt contains more than 200 million of sequences. Again, there are many model organisms that are underrepresented at rcsb.org. So for that reason, we think the integrating CSM, it's important for our users. Of course, that one could argue, but if there is resources of AlphaFold DB and other databases, why we should include them in rcsb.org? I understand, and I think that AlphaFold DB has done an amazing work, and it's a really amazing resource, but the richness of tools for visualization, for searching, for analysis that we provide at rcsb.org, you won't find them in any other place. So if we treat computed structure models in the same way as we treat experimental structures, and they are integrated in the same way, all these tools will become automatically available. So this is going to provide a really rich ecosystem for analyzing protein structures. So our current situation is we integrated the first version of AlphaFold DB, and that contain more than 1 million of protein models. And we have also important model organisms from ModelArchive. That is a repository where user can deposit their own computed structure models. So we have more than 100 million of computed structure models integrated at rcsb.org. And our idea is to grow up to the whole UniProt archive. So these 200 million. Another important question is, okay, so we have experimental structures and computed structure models. So when should I use a computed structure model? Well, this is not an easy question because I don't think there is a unique answer to that, and depends on your analysis. So what is the purpose? But as a general rule, we could say, if you have a good resolution experimental structure, 3 angstroms or better, that represent the whole structure that you're interested or a specific region or domain, then go for the experimental structure. Of course, that if there is no experimental data, the computed structure is going to be your best option. But you have to be careful here. Important, check the prediction confidence scores. We will talk a bit more in some slides. And the second thing that I would be quite careful is your analysis relies heavily on the position of the sidechain residues. You have to be careful because it's true these models are really confident predicting the main chain, but the sidechain residues sometimes are not as accurate. So for example, if you are trying to calculate a protein ligand affinity, I would be quite careful or to use a computed structure model. Of course, that there is a situation where decisions can be quite complicated. What happens when I am interested in a full chain structure, but some domains or regions of my protein are available as experimental structures, experimental entries at rcsb.org. So yeah, this is when things come really difficult to decide. And again, this depends on your use case. So it's not always easy. Okay, so now before going to the hands-on, so to the demo, let's explain briefly how to find them, how to find computer structure models at rcsb.org and what information is available. The first thing that I want to highlight, at rcsb.org, we have now an icon-based system to identify the type of entry. So we have experimental determining structures that are those that are coming from cryo-EM, X-ray crystallography, NMR, that are represented with this Eldenmeyer flask icon. We have what we call integrative 3D structures, that are those structures that combine computational techniques and experimental methods. Brenda gave a really cool seminar, I think it was 2 weeks ago. So if you are interested in this type of structures, go to our rcsb.org YouTube channel, and you will find the presentation there. So those are represented with this Jigsaw icon. Jigsaw piece icon. And finally, we have the computed structure model. So that is what we are talking in this talk. That is with this cyan color instead of dark blue and the screen computer icon. So when next to the protein or the structure entry ID, you find the screen icon, this means that you are in front of a computed structure model. So what information is available? The short answer is the same. As I said, we treat computed structure models or we treat experimental entries. So the information is the same. This is not exactly true because there is some data that is coming from the experimental information itself. And of course, those entries, the computed structure models, are not coming from experimental information. So all the validation data, validation report, this chart that we show about the qualities of the structure, the experimental quality of the structure, are not going to be available. But instead of the validation chart, that you will find is what is called the PLDDT histogram. So we will talk about what is the PLDDT, but basically is a confidence measure. It tells you how good is the model. Okay, let's talk about what is the PLDDT. So first of all, PLDDT comes from predicted LDDT. And the LDDT is the local distance difference test. And this is the measure that is being used in CASP for evaluating the models. So the reason why this metric exists is because if you think if you want to compare a predicted structure, predicted model with the native conformation, there are some situations as displayed in this picture. This picture is coming from the original publication of the LDDT, that even if in this case that we can see that this protein is composed of two globular domains connected, displayed here. So sorry, here is the model is our green protein, well, green, red color gradient protein. And the reference, or what we could consider the native conformation, is the gray protein. So there is no way that I can superpose globally both proteins. Even if, as we can see in B, both domains matches, if I try to superpose one, the other is not going to fit. So this is what is trying to solve the LDDT. It's a superposition-free score, so you don't need to superpose the structures. That what is measuring is the self-similarity of the reference compared to your model. So let's talk a bit about that. What is this thing of the self-similarity? So the LDDT is defined per residue of your model. So, and what is being evaluated, so this number is made up. It's a bit just to understand how the score works. So it's a bit more complicated than that. But if I take a residue, my red residue in the protein model, and I consider its environment, what I have to observe in the native structure or the reference is that the environment is the same. Even if the orientation doesn't match, what I want to observe is the neighbors of my residue in my model are the same as in the native conformation. So in this situation, that the residue has exactly the same neighbors, so the LDDT is going to be 100. So the LDDT is evaluated between 0 and 100, or 0 and 1, depends on how you want to consider. But if you give it as a 10%, so in that case, all the residue matches, so LDDT is 100%. So for example, in this case, that my protein model has one more neighbor than the reference, the LDDT is going to be 80%, because only 80% of the neighborhoods are conserved between model and reference, or the native conformation. So going back to what is the PLDDT, the predicted LDDT is evaluating exactly that. So it's a bit more complicated because, as in general, when you have a model, you don't have the native conformation. For that reason, you want the model. But I'm completely sure that all of you have used artificial intelligence agents at ChatGPT. I am saying that because behind the protein folding methods and ChatGPT relies the same type of neural network that are transformers. So it's the same type. But there is a key difference between the folding problem and the chat agents. The chat agents hallucinate. So whatever you ask, it is going to give you an answer. And the difference is that this is an active field of research. They cannot provide a confidence score. So there is no accurate way that ChatGPT, for example, can give you a confidence value of how sure is about the answer that it is giving you. And because of the nature of our data, the protein folding methods are able to do that. They are able to predict the LDDT, and it works pretty well. So if the LDDT is high, you can believe that the model is a good model. So in this case, the histogram, what is displaying is the number of residues that have PLDDT high, very high. No, what means is defined as a PLDDT greater than 90, a confident PLDDT that are values that are between 70 and 90, low PLDDT between 50 and 70, and very low, less than 50. Don't believe anything that is less than 70. So if your model contains a lot of low or very low PLDDT residues, don't trust this model. You can, in fact, these residues, you should consider them as unmodeled residues. So because the network, it predicts, so in fact, it's happening the same as the chat agents. So it's hallucinating. So it doesn't know what to say. So it just says something that, okay, this is my best, but I have no idea. And again, what I said before, the difference is that the network knows that it's hallucinating. So consider the low or very low as unobserved residues. Never use these conformations. Okay, so how do I find computed structure models at rcsb.org? Short answer, the same way as you search for experimental structure with important detail. So for example, if you used to search PDB entries using our main text search bar, you will see a button. So the only thing that you have to do is toggle on the button that says include CSMs. The same if you are using our advanced search tool, you have to do exactly the same. Next to the search button, before starting your search, you have to toggle on the include computed structure model switch. Okay, so let's do a small demo on these things. So if we go to our website, let's start by our main search tool, and we search, I don't know, ribosome. So what we can see is that, okay, all the structures contain the Elden-Meyer flask. So all these entries are experimental entries. So this office hour is not about advanced search, but please, if you have any questions, I will do my best to answer them. So on the left side, you have our drill downs that you can use to refine your search, to add other conditions. So in that case, all these entries correspond to experimental entries. There is close to 10,000 of experimental entries. Just to show you, if I choose the integrative models, what I see is the jigsaw piece icon here next to them. So again, if I want to include computed structure models, what I have to do is, let's do the same search, ribosome, and let's include computed structure models. Okay, so now here they are. Now, if we go again to our refinement panel, we can see now structure determination methodologies, 80,000 computational models. This is what I was telling. This is one of the reasons why we are doing this effort of integrated computed structure models. Look how many proteins were missing because we didn't have any experimental evidence from them. So this is what we win, including the computed structure models. So let's see what information is available here. As I said before, basically the information that we have is the same as for experimental entries. All the models will come with its structure summary page, where you have also the macromolecules section. You have the organism, you have genes, you can search by sequence similarity, search by 3D structure, and you have, for example, the structure tab where ModelStar will display the structure of the model. You have the annotation tab where you will find the gene ontology terms, protein domain classification based on Interpro, and so on. The information that we have is the same. What is different, again, is the validation report. So we don't have the chart that display what is the quality of the experimental structure, because there is no experimental structure. Instead, what you find is the histogram showing the confidence of the model, the PLDDT. So you will have the global. So this is the average among all the residues. So this is a good model, 95% of PLDDT. So it means that in average, all the residues have a 95% of the neighborhood is the same in the model that in the native conformation. If another useful tool to have more granular information on the PLDDT is the 1D and 3D tool. I gave office hour a few months ago. Again, if you are interested in this useful tool, go to our YouTube channel, rcsb.org YouTube channel, and you will find the seminar there. I'm saying that because this tool will facilitate quite a lot the mapping of the residue values from the sequence to the structure. No? So for example, I can click in one of the residues and I can explore locally what is the structure of the neighbors and the residues. In this case, as I said, this model is really good. It's a really confident model. So let's go back again to our initial search and let's try to search for a more tricky case. Ribosome, remember, you have to include computed structure models. I always forget and I always think, where are my models? So, okay, so this can be a tricky case. This case, so the average is 78%. 78%, it is not that good. Okay, I know it's higher than 70. That is where you can start trusting the structure, but depending on what is the purpose that you are using this model, it can be a bit, you have to be quite careful. So in this case, the sequence has around 300 residues, and we can see that more than half of them are defined only as confidence. So if we go to the 1D, 3D, yeah. So, okay, so we could more or less see that this protein has like 4 globular domains, more or less. No, one here, another here, another here, and another here. So if we see this region here, and I think this is one of the useful things to use the 1D, 3D. So you can see, oh, sorry about that. I wouldn't trust this fold. Even if it seems like a real fold with 3 nice beta strands and an alpha helix there. So it's something that one could, oh no, that can be a protein fold domain. So it's trustable. No, don't trust it. Because as I said, this should be treated as an observed. This shouldn't exist. So you shouldn't be using regions like that. The rest of the protein, as I said, if you are interested in analysis that don't require, don't rely heavily on the atomic position, as for example, you are trying to do some kind of interaction protein ligand, I wouldn't use this model because the confidence is not that good. Just to finalize this demo, let's go to the advanced search. And what I was saying at some point in the presentation. So if we integrate computed structure models at rcsb.org, all our search functionalities, and we have many of them, chemical attributes, sequence similarity, sequence motif, structure similarity, structure motif, you can use any of them and you can search computed structure model. So to give an example, let's choose structure similarity. Let's search by chains. And yeah, and again, what happened is what always happened to me. I forget to include the computed structure models. So it's the only thing that you need to do. And I don't see any model here, but remember, you can, if you are interested in exploring models in particular, you can always use our drill down to refine your search, choose computational. Remember, it's not enough only by checking the checkbox. You have to run again the search. Okay, and here they are. So Now I get, in that case, all of these looks really high quality models. You can see just because of the image, because everything looks really dark blue. So yeah, again, this is a pretty good model. So this, you can be quite confident that the fault is the right one. And just to end the demo, in the structure summary page, we provide many links to search, to advanced search. So I don't know, for example, we have organism. If you click here, what you do is you go to our advanced search and you are searching by the organism name. So when you are coming from a computed structure model, the option for including other computed structure models will be always available. So this is different when you are, so if this was an experimental entry, I use any of my, for example, search by enzymes, the computer structure models won't be active, but it is on in the case that you are coming from a computer structure model. Okay, so this is what we have done in the demo. So thank you, all of you, for attending to our office hours, and I will be quite happy to answer any question.